All right, for our last video for chapter 2, it's actually going to be a bit of a heavy one. We're going to be getting into the organic molecules, also known as the biomolecules or the macromolecules, where we're going to talk about the 4 major categories, namely carbohydrates, proteins, lipids, and nucleic acids. And we're going to get all into the details of each of those groups. So first and foremost, we want to define organic compounds. What do we mean by that? We don't mean organic like you would find in the grocery store, right? What we mean is we're talking about hydrogen and carbon and sometimes oxygen as well. But generally speaking, carbon and hydrogen must always be present in an organic molecule. That means that water is actually not organic, right? Because it doesn't have the carbon. It's only H and O. So organic, that's our strict definition. Now, we have 4 major categories of organic compounds. We have the carbohydrates, nucleic acids, proteins, and lipids, and we're going to get into each of these. So let's talk about what these hydrocarbon structures are. So hydrocarbons, this is just, as the name implies, something made of hydrogen and carbon in a very organized fashion, usually either in a chain or a ring. Right, so we're seeing a chain of carbons with hydrogens branching off of them, or we're seeing a carbon ring with those same hydrogens branching off as well. Hydrocarbons by themselves, like if it's just the hydrogen carbon, these are hydrophobic. But we have various functional groups that branch off of the hydrocarbons that can give very unique properties. This is getting into some organic chemistry, so we're not going to necessarily worry about memorizing all of the functional groups, but we do want to at least be familiar with some of them, especially a handful that are going to come back over and over. So down below we see a couple, we see ketones, we see organic acids, so COOH, right? Alcohol, or the OH, or hydroxyl, sometimes you'll see it called aldehyde, which is COH. Carbonyl, so here on the right we see carbonyl, we see hydroxyl, which is the alcohol, we see sulfhydryl, we see amino, we'll see that come back, we see carboxyl or carboxylic acid, which is a COOH, and phosphate, right? We'll see phosphates as well. So if there's like the ones you see here on the slide, these are ones we're going to talk about. Right? So these are ones we're going to want to pay attention to. I'm not worried about like the rest of them necessarily. The ones on the list here on the right, definitely, especially hydroxyl, amino, carboxyl, and phosphate. Those 4 we're definitely going to be talking about. We're going to see those guys come back over and over again. Now, let's go ahead and get into our first category of the 4, and that is the carbohydrates. These are usually water soluble, meaning it dissolves in water, organic compounds, and most of the energy that we use on a daily basis is in the form of a carbohydrate. So we are seeing CH and O, and we're seeing them, generally speaking, in a very specific ratio of 1 carbon to every 2 hydrogen to every 1 oxygen. So we see this very specific pattern, not just carbon, hydrogen, oxygen, but there's 1 carbon for every 2 hydrogens for every 1 oxygen. Most of the time we're looking at carbs in terms of their ability to function in the process of making ATP, right? And we'll talk about ATP a little bit later. We also can convert carbohydrates to fats or proteins when we have more carbs than we need, and this is why you don't have to eat a perfect ratio of carbs, fats, and proteins to survive. We're very much capable of converting these different organic compounds from one category to another if needed. We can divide the carbohydrates into 3 different groups based on the size and complexity of the molecule. The simplest is the monosaccharide. It's 1 single sugar unit, basically, which is C6H12O6. So we call it a monosaccharide, mono meaning 1, saccharide we think sugar. So this is a simple carb, and it generally tastes sweet, right? So like glucose, right? It tastes sweet. And that would be a good example of a monosaccharide. Disaccharides, di meaning 2, saccharide sugar, so we have 2 sugars together. So we basically taken 2 monosaccharides and glued them together. And so these are also relatively simple, pretty small, and these are also sweet. So like the sucrose or the table sugar that you use in like your coffee that is sweet, that would be a good example of a disaccharide. And then the last one is the polysaccharide. Poly meaning many, Saccharide meaning sugar. These are complex carbs. And we say complex, we're meaning hundreds of these sugar units, hundreds of monosaccharides all bound together. These are not sweet, generally speaking. These are more savory. So when you think of something that is like bready, that doesn't have that super sweet taste to it, or starchy, right, like a potato, that's where we think polysaccharides. So again, our monosaccharide, again, one, mono meaning one or single, we have one sugar, usually in a ring, so it can be a pentose or a hexose type sugar, so that just has to do with 5 or 6 carbons. And we see they're all pretty small and simple, right? So like something like glucose, it's C6O12O6, that's not too big, right? There's other ones like ribose, fructose, galactose, dextrose, deoxyribose. These are all examples of monosaccharides. The disaccharides, like I mentioned, sucrose, this is just table sugar that you would put into your coffee, right, or your tea or whatever. This is just 2 monosaccharides that have been bound together. And they're bound together by a dehydration synthesis reaction. Now I'm pointing this out because we're going to see this dehydration synthesis reaction come back over and over again, and it has the same basic process in that we are going to remove a hydroxyl group from one and a hydrogen from the other. So we're removing an oxygen hydrogen from one and the other hydrogen from the other one. So H2O. We're basically removing a water molecule to create these open links that can now link together. So we've removed water. So dehydration synthesis. Again, we've removed water to build up a larger molecule from smaller ones. Now the polysaccharides, again, we're looking at hundreds of monosaccharides glued together. So here's like glycogen here, right? Oh, massive, right? Absolutely massive in comparison. We don't even see the little rings anymore that we saw here. Right here is really easy. We could draw every single individual atom that was involved in that molecule. But here, oh my goodness, we're looking at hundreds. So glycogen is a good example of one we find in humans. This is how we store things like carbs in our liver as well as in our muscles. So glycogen, big one that we use here. Starches, cellulose, these are plant related. So starch is going to be like one that we get in our food, right, in our diet from plants that we eat. So like potatoes are a good example of a starchy food source. And then cellulose, this is the indigestible part of plants. This is what we would know as fiber, like dietary fiber. It's not something you can actually get nutrition from, but you do still need it because you need that bulk when it comes to eliminating fecal waste. Our next category is the lipids. Now, lipids are kind of, I like to think of them like the junk drawer of the macromolecules, simply because they have a lot of different types of structures to them, rather than how with the carbs we saw kind of a basic structure with that sugar, that carbon ring sugar that's just either bound, single, two, or a lot of them bound together. Lipids have some great variety in how they're structured, but there's one thing that keeps all of them together in one category, and that is their ability to be hydrophobic, right? So they will be hydrophobic because we have that hydrocarbon base. A lot of these lipids are going to be hydrophobic, or all of these lipids, I should say, are going to be hydrophobic. So that's kind of what keeps all the lipids together in one category. Now we're going to talk about a few different roles that lipids can play, and basically look at the different categories, the different types of lipids we see. So when it comes to long-term energy storage and protection, we see the triglycerides. When we look at cell membranes, we see phospholipids. And then when we see steroid-based or lipid-based hormones like testosterone and estrogen, right, each of these are going to have a slightly different molecular setup. So let's start with the triglycerides. The triglycerides, as the name implies, are going to be 3 fatty acids, that's where the tri comes in, bound to one single glycerol, right, glyceride. So we have one glycerol backbone and 3 fatty acids linking right off of that, like little tails. Now, these are going to be for protection, so we can use them as like our fat padding under our skin, insulation, same thing. to keep us warm. It can also be to store energy. So our body fat, right, is going to do all these things. It's going to store energy for later. It's also going to help insulate us if the weather is cold and provide protection against physical harm. So pretty simple in terms of their structure, and I'm not going to get too crazy in terms of knowing like palmitic versus stearic or oleic acid. I'm not worried about that so much. One thing that we do want to point out is saturated versus unsaturated. Saturated means that all of our carbons have single bonds, which means we can have the maximum number of hydrogens possible. But over here, like in this oleic acid, we see a double bond. One that's going to create a bend in the tail, and we don't have the maximum number of hydrogens, right? Because we don't have the one here or here. So two of them are now gone because this double bond is there instead. So this is why it's called unsaturated, because we do not have the maximum number of hydrogens compared to saturated, which does. Saturated tails, we see they're nice and straight. They can stack on top of each other very easily. Stack, 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 stack. So at room temperature, things that have high saturated fat content tend to be a solid at that temperature, right, at room temperature. Whereas an unsaturated molecule or an unsaturated fat tends to be a liquid at room temperature instead, because that bend in the tail, that little kink there, is going to prevent the nice flat stacking of molecules on top of each other. Now we can use hydrolysis, meaning using water to break triglycerides down, right, to form free fatty acids, which we can now use for energy, or we can break them into ketone bodies in the liver. Sometimes ketone bodies can be really elevated. So this, like if you have a really low carb diet, let's say you are on the keto diet, right? Where did keto diet get its name? Basically, if you have a low carb diet like the keto diet, you're going to see an increase in ketone levels because you're using fats for your primary fuel source rather than carbs. This is what we call ketosis. We also see this in uncontrolled diabetes. So they'll see a large number of ketones in the urine, right, or in the bloodstream, right, because of this. People who are doing the keto diet sometimes call like the first little bit the keto flu, and it's probably because of the high ketone levels that they're not used to. If you have a diabetic who is starving, we see very high ketone levels, which are going to lower the blood pH, which means we're in ketoacidosis. Ketoacidosis, or what's sometimes called DKA or diabetic ketoacidosis, is a medical emergency. Like, you need to get to the ER now. This can result in coma and or death, right? So if you have a diabetic who is not well controlled and is not able to get anything to eat, their ketone levels will go through the roof, and they will see acidic like an acidic pH in their blood to the point of damage. Again, medical emergency in that case. It's called DKA for short. Now, these fatty acids we talked about, again, are just these long chains of carbons with the hydrogens coming across them. And one of those ends is going to be a carboxyl group, or a COOH. That's going to allow us to link to our glycerol backbone. Again, saturated means we have single bonds all the way down, so we have the maximum number of hydrogens. Unsaturated, we do have a double bond, at least one, that causes a kink in that tail, and it prevents us from saturating the chain with the maximum number of hydrogens. Our next lipid is the phospholipid. The phospholipid is similar to the triglyceride, but we've modified it slightly. We still have a glycerol backbone, but we've modified it from 3 fatty acid tails down to 2, and that third fatty acid has been replaced with a phosphate group. Now, this phosphate group is critical here because this is going to be a polar end of this molecule. So the fatty acid tails are still nonpolar. They don't like water. But that phosphate head loves water. So now we have a molecule that's considered amphipathic, meaning, amphiphilic, meaning it likes both water and hates water. And because of this hydrophilic and hydrophobic sections on this molecule, it will spontaneously organize itself so that the phosphate heads are facing water and the hydrophobic tails are facing themselves. Right, so this is what's going to cause a spontaneous generation of what we call the phospholipid bilayer. And this is key when it comes to forming something like a micelle, which would just be like a little phospholipid ball, versus a cell membrane, right, which all of our cells use to create a barrier between the inside and outside. Our last category of lipids are the steroids. Steroids have a category or a characteristic that is unique to them, and that is the 4 fused carbon rings. So we see this same backbone. Let's see if these A, B, C, and D, these are the 4 rings. This is what we call cholesterol, right? These 4 rings together. So sometimes it's called the fused ring backbone or the cholesterol backbone. And then the difference between all these different steroids are just the groups that branch off. So like here we see a hydroxyl group, we see a hydrocarbon tail. Here we just see like a hydroxyl group and some methyl groups. Over here we have some methyl groups. We have carbonyl group, right? Hydroxyl group. Over here we just have a hydroxyl group. So we see all these just the same basics, but the branches are just slightly different. And so we'll see similar backbones among all of these. Cortisol, estrogen, testosterone, aldosterone, bile salts, vitamin D. But if we look at a lot of them, estrogen, progesterone, testosterone, aldosterone, we see similarities in the name, right? So keep that in mind when you're seeing the names of hormones. If you're seeing the same types of names pop up, like estro, testosterone, right? Sterone, we see that a few times. That gives you an idea of what we're looking at. Now our next category are the proteins. Proteins are normally water soluble. They can have some parts that are non-polar, but generally speaking, they will dissolve in water. And this is like our big category. When we talk about the function of proteins, the functions of proteins are vast, right? Proteins can be used for muscle contraction, to transport things in and out of cells, to transport things around the body. They can be various substances used to help speed up reactions like enzymes, protection. The list is really long. They're very diverse in what they're capable of doing. And the big thing we're introducing with proteins, besides the carbon, oxygen, hydrogen, is that we are going to be nitrogen heavy, right, in comparison to the others. So we're going to see a lot more nitrogen here than we saw in the other categories. So when we look at proteins, they have a basic building block, what we call a monomer. So it's like the single little unit called an amino acid. So the amino acid is the building block of proteins. There's 20 different amino acids, 9 of which are essential. The other 11 are not essential. What does the essential mean? Basically, if it's an essential amino acid, it means you must eat that amino acid in your food. You don't have the ability to manipulate it into that particular amino acid. So the other 11, you can use other things to make those amino acids in your body. You don't have to consume them necessarily. You can just build them from smaller building blocks. But the essential amino acids, you don't have a pathway to make it, so you have to get that amino acid in the food you eat. Most of the time, eating animal products will get you all of your amino acids. If you are vegetarian or vegan, you will have to pay more attention to what you eat to make sure you're getting all 9 of these essential amino acids. It's entirely possible to get them if you're vegetarian or vegan. You just have to be a little bit more mindful to make sure you're getting the right combination of foods to ensure you're getting all 9 on a regular basis. There are some complete proteins, meaning they have all 9 in a vegetarian or vegan diet, but it's much harder to come by compared to someone who has animal products in their diet. So these amino acids, even though there's 20 different kinds, they all have the same basic structure. And that is we have a central carbon right in the middle. We have an amino group, NH2, and we have a carboxyl group, COOH. So amino, carboxylic acid, amino acid, that's where the name comes from. The one thing that makes the amino acids different from each other is this R group, right? Also called the variable group, the side chain. So there's 20 different R's basically when it comes to the amino acids. Now, some of these are polar, some are non-polar, some have positive charges, some have negative charges. I'm not going to expect you to memorize all these necessarily, but we should be able to kind of figure that out basically, or recognize that there are different types of amino acids in terms of charge and how, like partial charge, full charge, no charge, right? This is important because this is going to assist with the folding of the protein. Right, so depending on the order of the amino acids and whether they're nonpolar, charged, polar, whatever, is going to impact the way that the protein folds. So we mentioned at the very beginning, proteins are super diverse. They can do all sorts of stuff. We're just going to talk about a few of the major ones. So they can be structural, like the collagen fibers, the keratin we find in our skin. Enzymes, there's a laundry list of enzymes that will help speed up chemical processes in the body. Most of the time you'll see them end in A's, A-S-E. Not always, but a lot of them do end in that A's. So like lipase, amylase, right? Those are all enzymes that will speed up the breakdown of certain compounds. We have contractile proteins like actin and myosin that are the major fibers, the major proteins we see in our muscles. We see them as signals, so there are protein-based hormones and cytokines that will signal and talk to other cells. A lot of receptors are protein-based, especially when they're embedded inside of a membrane. And then some of them that are also found in that membrane can be carriers or transport, so we can create a channel through a membrane to allow certain things in or out. We can transport things around, like hemoglobin can transport things around within the bloodstream. And this is just a short list. Again, we can even use them for like defense, right? So we can use like the membrane complex proteins that are involved in your immune system that basically punch holes in bacterial cells. You know, so there's a laundry list of things that proteins can do. Now, how do we form them? We use a dehydration synthesis reaction. Bam, we're basically removing water again to pull small units, the amino acids, together to form longer chains. In this case, we're calling our long chains polypeptides. Poly meaning many, peptide protein or amino acids. So we form this peptide or this polypeptide, right, by removing water. Now, the process of making these proteins, of how we link these together, is called translation. It occurs at the ribosomes. So the ribosomes are like the machinery that allow for us to do this connecting process. We're not going to get into the details of translation here because that's just not for this chapter right now. Now, remember how I said that I did not expect you to memorize all the different amino acids and whether or not they're nonpolar or polar or anything, but I did mention that we should recognize the importance of polar versus nonpolar versus charged amino acids because proteins are very particular about their shape. And so there's 4 levels of structure with proteins. We're going to go through them and then we'll talk at the end about why it's important. But number 1 is primary structure. This is just like the basic order of amino acids. This can be from 3 to 4,500 amino acids long. So these are huge, right? So if we think of primary structure of a protein, think of if you were to unwind the entire protein and string it out like garland or like a necklace, right? Like a pearl necklace. That would be the primary structure. Just what amino acids do you have in that long single strand? The secondary level of structure is the localized folding. So the alpha helices and the beta sheets that we see through those hydrogen bonds. Hydrogen bonds, again, are going to form when there's partial charges, so you know that you have to have polar amino acids in these sections. You can't have the nonpolar ones there in high amounts, right? The tertiary structure is going to be the overall 3D structure of a single chain, right? So we're looking at the entire long chain. What is its 3-dimensional shape? And then quaternary, this is optional. It depends on the protein. It depends. Like if, let's say we're looking at hemoglobin, hemoglobin has 4 different chains working together. So hemoglobin would have a quaternary level of structure. So hemoglobin, insulin, right? Insulin has two different chains that join them together. That would be the quaternary level. We're looking at how all the chains of the protein are linked and interacting together. So again, primary, think just the single string order of amino acids. Secondary, we think localized folding, the alpha helices, the beta sheets. Tertiary, we're looking at the entire chain in its 3D shape. And then quaternary, we're looking at multiple chains interacting together. Now, some proteins have other parts that have combined with them, like a protein with a carbohydrate. We call that a glycoprotein, or a protein with a lipid. That would be a lipoprotein. And we see a lot of these in the cell membrane. So these can be things that help us like anchor, help us connect, help us signal, help us receive information, right? Whatever. We'll get into those things as we get there, but these are just things we're going to find within that, that cell membrane. And now finally our last group, the nucleic acids. So nucleic acids, we're going to mainly talk about two of them, and that's going to be deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Here we're seeing the carbon, the hydrogen, and oxygen, as well as the nitrogen, but we're introducing another main player that we have not seen yet, and that is the phosphorus. Namely because we're going to have phosphate groups. Nucleic acids are built using the monomers called nucleotides. And nucleotides have the same basic shape. We have a pentose sugar, like deoxyribose or ribose. We have a nitrogen-containing base, and we have an inorganic phosphate group, or PO4. Those are the three main parts of a nucleotide. And the nucleic acid is made when we link multiple nucleotides together. So if we look at DNA, DNA is going to form our genetic code. We have the same basic structure no matter which person, animal, organism we're looking at, right? It's going to be double-stranded. It's going to have a double helix or a spiral staircase shape. And we have four basic bases we find there. We have adenine, thymine, guanine, and cytosine, and a deoxyribose sugar. Those bases, A, T, G, and C, right, for short, are going to bind in very specific ways. A's with T's, G's with C's. Now, RNA is a little bit more diverse. We have different types of RNA, like rRNA, tRNA, mRNA, microRNA, snRNA. I mean, there's a laundry list. We're not going to get into all of them necessarily. We were probably really going to just care about rRNA, tRNA, and mRNA. But these are single-stranded. They don't have a helical shape anymore because they don't have the second strand. And we have a lot of the same bases, adenine, guanine, cytosine, but we've replaced the thymine with uracil. And so in this case, our base pairs will be A with U and G with C still. So DNA, we have two different types of DNA in our cells. We have nuclear DNA. This is what you probably are thinking of when you think DNA because it's what we find in the nucleus. It's this, complex, it's basically like chromatin, which is the DNA with the proteins. We have the chromosomes. You get one set from your mom, one set from your dad. It's pretty straightforward. But we also have mitochondrial DNA. It turns out our mitochondria actually have their own DNA separate from ours. This is going to be inherited from your mom, right? So this is something inherited maternally. We actually can use this to trace things, right? Because we can trace down the maternal, basically lineage using mitochondrial DNA. So if we look at DNA in general, right, they all have the same basic structure in that we have two strands. The backbones of the strands are made of alternating sugars and phosphates. And then those nitrogen-containing bases are going to face the middle. And they're going to bind using hydrogen bonds, right? And we're always going to have the same type of pairing. Cytosine with guanine and adenine with thymine. So C's and G's, A's and T's always. You will not see G with T. You will not see C with A. If you do, that means something went seriously wrong. Now, on here we see the backbones called the rails, right? Which is just the sugar and the phosphate. So you might see them called rails. You may also see them called backbones. I'll call them backbones. The rungs, like the middle parts of the ladder, are going to be the bases. Again, we have our complementary base pair rules, A with T, C with G. Now, RNA, again, is a single chain. We don't have that second strand. We have lots of different types of RNA, including precursor mRNA, mRNA, tRNA, and rRNA. mRNA is basically what's going to be produced by transcription and is modified into a code that's going to be used to make a protein. Transfer RNA is going to bring amino acids for that protein making process, and then rRNA is going to be part of the ribosome, and that's going to be used to basically as the machinery to make the proteins that we need every day. Another special molecule we see, ATP. This falls in the nucleic acid group as well, even though it is relatively simple, but basically what we have here is we have, so ATP, adenine with a ribose, so together we call this adenosine, and then we have phosphates, namely 3 phosphates in a row. So that's where the name adenosine triphosphate comes from. This is a very special nucleotide. It basically serves as our energy currency molecule because it can store a lot of energy, and I mean a lot of energy in those phosphate bonds. You see them shown with these little little wiggles, right? These little squiggly lines. That is because these are bonds that are unstable but have a lot of energy in them. So ATP is a molecule that will not last very long. You know, we generally keep like a few seconds or so worth of ATP at any given moment and store long-term energy in the form of carbs and lipids because of their stability. But ATP, even though it's unstable, is massively helpful in terms of powering other chemical reactions. So huge amounts of energy, especially the further down the chain you get. So releasing just one of these phosphate groups can power another chemical reaction very easily. And we'll see that happen going from adenosine triphosphate to adenosine diphosphate over and over again. So huge amounts of energy between those phosphate groups. And so we have entire chemical processes dedicated to breaking down things like carbohydrates and lipids and proteins and whatever to produce this ATP, right? We're always making more ATP to form these high energy bonds. We'll talk about cell respiration in the next unit so we can get into the details of how we make all these ATP molecules. But for right now, we just at least want to have in our head that ATP is this very powerful, very high energy molecule that is used throughout all of our cells.